Hello everybody! Today we are going to be talking about variation of heat shock time in the transformation of E. coli cells. Today's presentation is brought to you by Caitlin, Avery, and Trent.
Transformation is the incorporation of foreign or exogenous DNA into a cell. What it does is it alters the genetic makeup of the cell by adding DNA from a different species. In this lab, E. coli cells are being transformed. Plasmids are being used to carry this out. Bacterial DNA was used to do this, or otherwise known as plasmids. The genes that were carried via the plasmids were ampicillin resistance gene and the GFP, or green fluorescent protein gene. The ampicillin resistance gene did not physically change the cells, while the GFP gene made the transformed bacteria glow green under UV light and then appear green or yellow in white light. In the protocol, heat shocking the bacteria causes it to take up the plasmid from the surrounding solution. The impact of changing the time, whether increasing or decreasing it, is sure to be exhibited in some way on the plates due to the fragile nature of the bacteria. This being said, from our experiment, we reduced the heat shock time to 30 seconds which we hypothesized would increase the efficiency of the transformation. Our reasoning behind this lied chiefly in our belief that the bacteria would be allowed to have a greater rest period before being put back on ice with only 30 seconds rather than 90 seconds in the heat shock, thus improving the transformation efficiency rate in comparison in last week's experiment. For this lab, we looked at two articles to back up our hypotheses, and in our first article titled Plasmid DNA Transformation in E. coli, in this article, it provided graphical evidence that a 30-second heat shock was more beneficial to the transformation efficiency than a 90-second one was. Going with this, in our second article that we looked at, titled Transformation of Plasmid DNA into E. coli Using the Heat Shock Method, in this article, another time comparison between 90 seconds in heat shock was examined. We used their results as a source of info for a different time frame besides 90 seconds to compare and contrast between these articles and our data.
We determined our hypothesis mainly off this graph from the first article because, as you can see, the 30-second heat shock has a higher transformation efficiency than the 90-second. Our results, as shown, all the LB plates had long growth. The only one that had no growth was the LB-AMP11 that did not have the plasmid in it. The other plates, both the plus plasmid and the plus plasmid plus variable, they both had similar results. The 1-to-1 had too many colonies to count. The LB-AMP 1 to 10 had several hundred. The 1 to 100 was less than 100, and the 1 to 1000 was only a handful of colonies. As you can see, these are reflected in the images. The top two are the growth, and then long growth, then too many to count, and then it just keeps decreasing and decreasing in colonies. The top two on the third column, there's a bit of a glare there, so they are in the picture off to the right, which shows the long growth and the no growth. So for calculations, we had to calculate the transformation efficiency and the fold difference. We calculated the transformation efficiency off of the 1 to 10 plate. And so for the normal plus plasmid plates, the transformation efficiency was 5.44 times 10 to the 5th colonies per microgram per milliliter. The transformation efficiency for the variable with plus plasmid was 8.1 times 10 to the 5th colonies per microgram per milliliter. And then we calculated the fold difference, which ended up being 0.49.
And based on our findings that Caitlin just went over, there was not a significant difference between the transformation efficiency between the times of 30 seconds and 90 seconds. And just looking at the Petri dishes, it didn't look like there was a noticeable difference. And then once we looked at the full difference, and which shows if there's a significant difference between the two, we noticed that our FD was 0.49. And this causes us to reject our hypothesis that if heat shock is reduced from 90 to 30 seconds, then the transformation efficiency will increase. And that's another beautiful hypothesis coded by an ugly fact. And then here's our two references, which Caitlin and Avery referenced earlier in this video.